For thousands of years, humans have been admiring the beautiful night sky. Early astronomers took this admiration one step further when they noticed that these bright points of light moved. Through meticulously tracking and measuring these changes, they were able to discover planets and build our modern model of the solar system. My research uses these same scientific techniques, but instead of looking through a telescope towards the heavens, I turn my microscope towards the universe within. My night sky is the living genome. By combining particle tracking techniques that these early astronomers used with fluorescence cross-correlation spectroscopy, a biophysical technique of measuring changes in brightness over time, I can collect data on the genome in action. From these measurements and a little bit of math, I can begin to answer questions like, how often does a particular gene turn on? What's triggering it? Or my most recent project, which looks at how the common COVID drug, dexamethasone, is impacting the genome. All of this promises to shed light on human disease and lead us closer to personalized medicine. These measurements are taken with a custom-built microscope here at Boise State. It's one of just 5 like it in the whole world. This microscope, known as the 3D orbital tracking microscope, is unique in that it is able to image for longer and faster than traditional microscopes. It's gentler too, so no longer do I have to image dead or fixed cells. I can watch live cells in action. All of this is done by, in essence, ignoring the edges of the image and focusing all the microscope's power on that point of interest. The image on the right, which I took with this microscope, showcases the nucleus of a cell. It's the bright green circle in the middle. Inside the nucleus that houses the genome, there is a single bright point of light. That bright point of light is an active gene. It's, in essence, my planet. By tracking this bright active gene, I'm able to begin piecing together a model of how the genome behaves. The human genome was sequenced in the early thousands, and while it was enlightening, it didn't answer all our questions. We have the code or the letters that make up the genome, but we don't understand the rules or guidelines that it's following. My research focuses on filling this gap. By collecting data on the genome in action, I can begin to build models, and along with it, our understanding of human health and what makes you, you.